everybody. My name is Dori, and I am a second-year PhD student at Durham University, and I'm also part of the MOSMED, and I work with the CCDC, and my project is about how Jason mentioned about going from fragment screening to fragment growing and new strategies for drug discovery. So my project focuses on glucokinase, which is a 52 kilodalton enzyme with 465 residues. And the reason why I'm focused on this enzyme in particular is because it plays an important role in the body. In the liver, glucokinase converts glucose into glycogen, which acts as a form of energy storage in the body. And the way that it kind of works is that at first glucokinase is bound to a regulatory protein. And as glucose comes in, the regulatory protein unbinds and glucose is changed into glucose 6-phosphate. Then it is transformed into glucose 1-phosphate and enters the cycle, and eventually it becomes glycogen in the liver. It also plays an important role in the pancreas, where it is involved in glucose-stimulated insulin release and therefore helps regulate and maintain glucose homeostasis. Its structure is made up of two domains. One is a bigger domain and the other one is a bit smaller. And it also has a flexible hinge region that allows it to undergo needed conformational changes depending on what it is bound to. So the crystal structures that we have solved, that are solved in the PDB, exist in 3 different conformations: in the closed conformation, in the open conformation, and the super-open conformation. So when glucokinase is bound to glucose, it is in its closed form, in its active form. And then as glucose gets transformed into glucose 6-phosphate, it opens up. And there it's still in the active form. It can either bind to glucose again, or it can open up completely and become inactive. So super open form is its apo form, and that's where it's in the super open conformation. This can also be seen in the crystal structures, where it is very clear how it kind of opens up, and in the closed and the open, it's bound to glucose. In the super open, it's not bound to glucose. The reasons why there's a lot of research into glucokinase is because of its clear link to type 2 diabetes. And this is because it's involved in the glycolytic pathway. So being able to activate GK can lead to the control of the glycemic imbalance that is associated with type 2 diabetes. And this is especially important nowadays with the rise of diabetes cases. In 2015, approximately 400 million people were diagnosed with diabetes, of which 90% was type 2 diabetes, and this number is still rising. We do have current therapeutics, obviously, but these can often have adverse effects, including hypoglycemia, gastrointestinal disturbances, and also weight gain, which is obviously something that we don't want. And so because of this, there's a lot of research into glucokinase activators that could activate the protein. And these tend to be small molecules that bind to the allosteric site, stabilizing the high affinity conformation and activating glucokinase. An example of a activator is dorzagliatin, which is currently in clinical development, and it targets both the hepatic and pancreatic glucokinase, which is very important because we want to target both. There are different types of activators. Some only target the hepatic glucokinase, but targeting both is preferential. Here are some examples of glucokinase activators from the PDB. They all look kind of different, but they all share the aromatic rings and a variety of elements, including sulfur and fluorine. Yes, so when it comes to my project aims and my project started with lab work and optimizing glucokinase expression and purification so that I could make protein that I could later on use in protein ligand binding experiments to see how glucokinase binds to different types of ligands. And then also this was done to aid me in glucokinase crystallization to determine the crystal structures of protein ligand complexes. In parallel to my lab work, I also work with molecular docking, and using gold in particular. And this is to kind of be able to run experiments to dock the protein with its ligands and then be able to compare this to my results in the lab. And overall, I'm trying to identify potential molecules that could show promise in glucokinase activation. So when it comes to the optimizing protein expression, I designed 3 different vectors, codon-optimized vectors, to allow me to express glucokinase. I performed expression tests for these, and these can be seen in the gels right here. At the start, I varied different factors, including the time of expression, the temperature, and then whether I used IPTG to induce or not. And as you can see, the protein is around 52 kilodaltons in size, so it's located around here. And for all expression tests, I got my protein to express. The time and the temperature didn't really seem to have that much of an effect, but IPTG had to be used to get a bigger, higher yield. And this allowed me to choose my conditions for the expression and for large-scale expression, after which I made a lot of protein, but I had to purify it to get only my protein, and I did this by using affinity chromatography, using a Hystrap HP column. This is an example of one of my gels for the first vector. The first row is just the ladder. Then we have the S, which stands for supernatant. So that's the protein before purification. AC is after column. So after the protein was loaded onto my column, and then F is just the flow through. And these are my elution fractions after the first purification step. I got a lot of protein, which was really good. The yield was high, but it was impure, so I had to continue with a second purification step. And I did the size exclusion chromatography using a SuperDex 200 column, and I was able to purify the protein further. There are some proteins here, but overall these fractions on the right-hand side seem pretty pure. I did the same kind of thing for the other 2 vectors as well. Those worked pretty nicely as well, and I was able to get a lot of protein at high yield. The identity of the protein was determined by MALDI mass spec and then also electron spray mass spec to make sure that it was glucokinase, and it was glucokinase. And I also performed CD to prove that my protein that I was getting was folded. Continuing from then, I worked on thermal shift assays, and a thermal shift assay is a method used to characterize and determine the thermal denaturation of protein samples, and it works by employing a fluorescent dye, which fluoresces in hydrophobic environments and undergoes rapid quenching in polar environments. So you start with your protein in its folded form, and as you increase the temperature, your protein unfolds, you get a fluorescence peak, and that helps you determine what the denaturation temperature of the protein is. And the 2 main uses, or what I use thermal shift assays in this case, I used it to determine what conditions help my protein be stable. So I ran a salt screen, an osmolyte, and a pH screen to determine what conditions I could use later on for crystallization that would allow me to get good crystals. And I also determined the denaturation temperature, which is around 43.5 Celsius. And then the other reason and the other experiments that I ran with thermal shift assays is in collaboration with Newcastle and Celine Cano's group there, because they have 24 potential activators that they have proved that they show some kind of activity. And so I ran the TSAs with glucokinase and these ligands to be able to find if the ligands have any kind of stabilizing or destabilizing effect on my protein. And I also varied the compound concentrations to see the effect of concentration on the effect. This is an example of my one of my TSA results. It's pretty complicated. So in A1 to A3, it was done in 96 well plate, and in A1 and A3, I only had the protein, and that helped me. The average of the 3 measurements allowed me to get the denaturation temperature. Every measurement was performed in triplicates to get a good representation, and then going from A4 to A6, that's this compound at 500 micromolar, and so on, continuing down the row. And the colors indicate the stabilizing effect of the ligand. So for example, the blue means that because the temperature increased compared to the denaturation temperature of just the protein, that means that the ligand had a stabilizing effect. And then the yellow and red mean that the temperature decreased, so the ligand had a destabilizing effect. When it comes to the results and the observations, I observed that generally glucokinase has a pretty low melting temperature, around 43.5 Celsius, which is relatively low compared to other proteins. And then also the delta T values in many cases were negligible. So for example, for B1, when the temperature there was 43 degrees compared to 43.5, that's just a 0.5 degrees of a difference, and that isn't a lot. And also overall, there were no clear trends that I saw from my TSA results. So for example, if you have a stabilizing effect of the ligand at a low concentration, you would expect it to have a stabilizing, even more stabilizing effect at a higher concentration, but that really wasn't the case. And I attribute it to compound solubility issues because it was really hard to dissolve the compounds, and I had to use 100% DMSO, but obviously in the well then that is diluted. So it's very likely that the compounds crashed out. And doing these experiments proved to me that using TSAs for my particular system and my compounds was not viable. So in parallel to my lab work, I also work on molecular docking and fragment-based methods. So fragment-based methods are basically methods that utilize fragments to develop molecules that can be used as potential drugs. And in the future, I'll be working with fragments to dock them and grow them into potential drugs with a collaborator. And I also work with molecular docking because it plays a role in the FBDD process. It is used to predict fragment protein interactions and their binding modes. And in the beginning of my computational work, I worked on doing analysis on my target protein, and this was done by scanning 24 PDB entries to identify entries that would represent my protein system and its different conformations. So because glucokinase is made up of, has 3 different conformations, it is changing, and I had to find PDB entries that would be able, that I could study, that would represent my system. Out of the 24, I chose 5 and narrowed it down to 5 based on mainly their resolutions. And I chose 3 entries of the closed conformation, 1 of the open and 1 of the super open. And this was because in most of the cases, activators bind in the closed conformation. So there's a lot of emphasis on that particular form. Because my protein has the different conformations, I'll be doing ensemble docking to show the way that the protein works. And to do this, I need a selection of proteins to work with. So to prep my system for ensemble docking, I redocked all the crystallographic ligands in gold into the protein to kind of figure out how I had to optimize the protein to be able to recreate the crystallographic ligand. And the optimization includes determining which active waters help with the binding, if there are any active waters, what tautomers I had to choose, and then also which hydrogen bonds play a role. So in most of the PDB entries, the protein, arginine 63 in the protein creates a hydrogen bond with the ligand, which was a very important interaction. And when I redocked the ligand for 3F9M, I was able to recreate it with the optimizations that I chose, and that was really good. The RMSD value was at around 0.2, which is good because I wanted to be below 1 to have a good fit. Then I did the same for 3IMX, and again, I was able to recreate the crystallographic ligand pretty well in gold, except for maybe this part, but still the RMSD is below 1, so that was good. But then for 3VEV, gold predicted a different pose to what the crystallographic ligand suggested. It suggested that the nitrogen should be oriented the other way around, which was unexpected, I guess. And to kind of research or like find out why this was happening, it was docked. And also, yeah, I forgot to mention, gold also suggested that a hydrogen bond should be present where the way that the nitrogen was oriented in the docked ligand, but this was not seen in the crystallographic ligand. So mogul analysis was done to see why this was happening, because a hydrogen bond in that spot would definitely be preferred. And so the mogul analysis works by just scanning very similar molecules and seeing if the bond lengths and bond angles are viable. And this was done for the crystallographic ligand, and it suggested that the angles that were in the ligand were not really within the viable range. And that means that the ligand was probably assigned not very well, and it meant that I was able to move on from it because the way that I was optimizing my system and using gold, the gold was doing a good job. I also docked the same thing for 4-ISCE, which is the open conformation of the protein. And again, the nitrogen was oriented the other way around, but this was using all the default settings and ChemPLP in gold. So once it was redone with ChemScore, the nitrogen was oriented the right way around. And I forgot to mention that for the 3-EVE VEV, I also did ChemScore, but redocking with ChemScore didn't change the way the nitrogen was not oriented the right way. And going on forward from this, now that I have my system set up, I can do ensemble docking, and I'll also be doing an evaluation of the scoring functions to find which scoring functions I could use that would suit all the different entries. Yes, and then to my current and future work. Currently, I'm working on using gold to dock the ligands provided by Newcastle to determine their viability. And then I have converted SMILES into 3D structures via RDKit and CSD Python API. So that's ready to go whenever I can. And I'm also setting up crystal trays to optimize the crystallization of GK expressed and purified from the designed vectors. And because my TSAs weren't very viable in the future, I'll be using surface plasmid resonance to study the binding of the Newcastle ligand to my protein and then comparing them to what gold suggests. So yes, that's it from me. Thank you for listening. And I would like to thank my lab group, my supervisor MK, and also Francesca from the CCDC for always being very supportive and helping me out in whatever I need.
Thank you, Dori. That was a super talk. So an obvious thing, talking about the crystal structure at the end there, where you found the nitrogen, the gold predicts the nitrogen the other way around.
Mm-hmm.
Have you tried getting MK perhaps try to re-refine the structure from the raw data, the crystal structure, and see whether actually you can place it the other way around as a sort of improvement?
Yeah, I haven't tried. We haven't tried, but it's a good idea. That would be really interesting to see what comes up.
I suspect it'd be a fun learning exercise as well to learn about the infelicities of crystal structure refinements and stuff too. Well, in that case, thank you. We'll thank Dori again.